So in this video, we are going to be looking at the genetics of multifactorial traits. If we go back to basic genetics, Gregor Mendel is known as the father of genetics. However, Victor McCusick is the father of human genetics. And as one has studied the genetics and as new areas of genetics have emerged, it has become apparent that multifactorial conditions exist. In other words, it's not simple Mendelian genetics anymore. Diseases such as type 2 diabetes and macular degeneration, heart disease, cancers, bipolar, stroke, I've got a whole list of these. There's more than one gene or one factor that attributes to an individual inheriting this condition. So, in actuality, few, if any, genes act alone, such as Mendelian genetics, which is what you would learn in biology and anatomy and just the very introductory level of genetics. Environmental factors can play a role, and then genes can be modified for expression. So what we're actually looking at is that most traits can be described as being more polygenetic or multifactorial in nature. So when we look at a single gene trait, such as big T is tall, little t is short, this is often produced as an all or none effect. They're either tall or they're short. Polygenetic traits, however, produce a varying phenotype, and so we call these quantitative traits. Our DNA sequence involved in these, we call these quantitative trait loci, or QTLs. So an example of a single gene disorder is cystic fibrosis. Now, we can have diseases that occur because of the genotype of the disease. They can be because of environmental influences. They can be because of modifier genes. So they can be polygenetic. So some common Snips that contribute small amounts of risk for a condition to show that could include hypertension, heart disease, osteoporosis, and diabetes, meaning environmental conditions could influence this. You have more than one gene that can affect this, and then depending on what SNPs that you inherit can determine how severe of the condition that you could exhibit. So polygenetic multifactorial traits are influenced by the interaction of genes and by the environment. Height, skin color, body weight, fingerprints, behavior are all examples of these polygenetic multifactorial traits. So let's go through an example of how this works. Lung cancer is caused by smoking, but lung cancer can also be caused by multifactorial traits that one inherits. So that an individual could get lung cancer without these multifactorial traits just by smoking, or an individual could get lung cancer, never have smoked, but have multifactorial traits within their body that causes them to develop lung cancer. Migraines are also multifactorial. If an individual has migraines, and my daughter does, my oldest daughter, when they have the migraines, if they have sensitivity to sound, then that factorial trait is coming from chromosome 1. If they have a pulsating headache and light sensitivity, then those multifactorial traits are coming from chromosome 5. If they have nausea and vomiting, then those traits are coming from chromosome 8. If they have a pulsating headache, sensitivity to light, nausea, and vomiting, then you have chromosome 5 and 8 causing the migraine issues. And so that's how we can start looking at how multifactorial affect an individual's phenotype. Fingerprint pattern is largely determined by genes and prenatal contact, with males having an average of around 145 total ridge counts, while females have a total of around 126. Height is another example of multifactorial traits that exist. Height is another example of a multifactorial trait. If we were to look at how the height has changed from, say, 1919 to 2019, one of the things we would see is that men have increased from 162 centimeters to 171. For women, they have increased from 151 centimeters to 159 centimeters. And so people are taller today on average than their ancestors from 100 years ago. And this is true for every country in the world. And it's believed that this is a trend that is due to general improvement in health and nutrition over this time period. And so we can see those multifactorial traits working in to cause us to be much taller than our ancestors were. Skin color is another multifactorial trait. Melanin is what protects us against damage from UV radiation and exposure to the sun. We have the same number of melanocytes per unit area of skin. However, we differ from person to person in the melanosomal number, the size, and the density distribution, which causes the varying colors of skin color. Now, testing DNA would indicate an individual's ancestry, and biologically speaking, it makes more sense to test the DNA to fine-tune and pick out which medical treatments would work best for an individual rather than going on skin color. If you go by skin color alone on a population level, then it would make sense, but on an individual level, it's going to cause errors, and it's going to lead to wrong medical treatments for individuals. And here you can see the key, the genomic key for the varying types of skin color and what genes they would have to inherit to get each of these skin colors. So the definition of race based on skin color is a social construct. It's not a biological concept. Skin color is not a reliable indication of a person's ancestry. Overall, 93% of the varying inheriting traits are no more common in people of one skin color than they are of another skin color. So, for example, cleft lip is not more common in one skin color versus another, but it is more likely to be in a person who has a relative with the condition. So you're looking at that genetic condition, not the skin color. So let's look at the risk of an individual inheriting a cleft lip. In identical twins, the risk is 40%, that if one twin has a cleft lip, the other could have it. In siblings, if one child is born with a cleft lip, then there's a 4% chance that their sibling could be born with that. Niece and nephew, 0.8%. First cousins, 0.3%. General population, not looking at relatives at all, 0.1%. So we've talked about multifactorial traits and how they could be due to environmental variance or could be due to genetic variance. Heritability is the estimation of the proportion of phenotypic variations in a population that's due to genetic differences, not environmental. And here I posted a chart of some of the human traits and the heritability of these traits. So to estimate heritability, researchers use several statistical methods. One way is to compare the proportion of people that share a trait with the proportion that's predicted to share a trait. And then another one is by comparing blood relatives and looking through blood relationships of individuals and seeing how a trait is passed from one family member to another, or how many in a family have a particular trait. So coefficient of relatedness is the proportion of genes shared between two people related in a certain way. So we could look at the likelihood of individuals sharing genes for colon cancer and getting colon cancer. We could say that the parent has it, and so then we can look at the degree of relationship. For example, right here, 1 degree separation. Then we could look at a 50 to 1/2. Siblings, a sibling has colon cancer, has the genes for that. Then we can look at the degrees. You can look at it from grandparent to grandchild, and then from cousins. So many times, when you look at risk factors for different types of cancers, you'll see that they'll give you these risk factors, these coefficients in there of, if your parent has it, then you've increased the percentage of possibly getting cancer by this amount. If your sibling has it, and then they go by the degree of relationship within the family. So if we're looking at adopted individuals, the adopted individual and the adopted parents, the similarities there is environmental, where if we looked at the adopted individual and their biological parent, the similarities there is completely genetic. So it's good to look at information on both sets, and that can help determine heredity versus environment for various traits. Twin studies are also very popular, especially if twins have been put up for adoption and each has been adopted by a different family. Now, when you look at twin studies, you have to look at dizygotic twins. You have to look at monozygotic twins. Dizygotic is going to be not identical. Monozygotic, they're going to come from one cell that is split. They're going to be identical. And so for a trait largely determined by genes, you would expect to have a higher connection between monozygotic twins than you would dizygotic, or a higher connection between identical twins versus fraternal twins. And so here I've put down a chart that shows you some different conditions and then the values associated between identical twins and fraternal twins. So let's look at an example like Alzheimer's. Look at that connection. If one of the twins has Alzheimer's, 78% chance the other one will, where if they're fraternal twins, you're looking at only a 39%. So separating out genetics and environmental influences, your fraternal, your dizygotic twins share environment and 50% of their genes. Monozygotic twins are going to have the identical genotype and they have the shared environment. Twins raised apart have the genotype, not the environment. Adopted individuals have the environment, not the genes. Now, I do want to point out that your dizygotic twins and just a sibling pair would share the environment and 50% of the genes as well. So the Genome-Wide Association study is used, they set this up to sequence the human genome, and then they started identifying Snips, and by identifying SNP patterns, that shows that individuals that have these same SNP patterns are more prone to have certain conditions. And that's really the direction that medicine is going, is by identifying these SNP patterns that will give indications of likelihood for various diseases. And then on the flip side of that, identifying these SNP patterns and what medication responds best with each SNP pattern, so that as we move on, medical treatment is going to become more and more personalized based on your SNP patterns within your genetics. So again, the Genome-Wide Association study is looking at SNPs that are shared with much greater frequency among individuals with the same trait than with other traits. And so that will help them connect SNPs with specific diseases and Snips with certain medications that work better. Now, this is going to save a lot of time and a lot of lives, because if an individual has a specific SNP pattern and there are 3 medications out there, you find out which medication that SNP pattern responds to the best, instead of going from, well, we'll try this first chemical. If that doesn't work, then we'll try this next medication. If that doesn't work, then we'll go to the third. Instead of wasting valuable time, you can immediately get the correct medication for the correct genetic code. Okay, so just a couple of reminders. Our DNA, we have roughly 3.2 billion base pairs, A, T, Gs, and Cs. And I keep talking about Snips. Remember, Snips are single nucleotide polymorphisms, easier to say Snips. And these are sites on the DNA genome where the DNA bases vary in at least 1% of the population. So in these studies, they take the Snips, they span the genome, and they look for these differences, and they find out, okay, all people that have Alzheimer's, they tend to have these SNP patterns. And then that helps us to identify likelihood of having a specific genetic trait show up. Now, it's not just the DNA, it's also environment. So you may have the SNP code for having a certain type of, say, diabetes. But if you exercise, you eat well, the environmental aspects you could control, that might limit the SNPs causing the DNA to progress. Now, with all types of studies, there are limitations. With the genome study, there's many data points, so that there are conditions of error. We also have associations between 2 types of information, but we don't have causes. We can have biases. We can have accuracy affected by complicated factors such as phenocopy or epistasis. We can also, because the genome is so large, we may miss very rare SNPs that would be important to identify for causative connections with certain diseases or disorders. Body weight is another multifactorial trait. About 30% of the U.S. population are obese, and about 35% are overweight. Now, we measure this with body mass index, and you take your weight divided by the height, and that will help give you your body mass index. And we are finding more and more SNPs that are associated with conditions of being obese and overweight. However, we also have to look at environmental conditions, and if we compare the U.S. to other countries, we have larger meals, we have a lot more fast food, a lot more processed food, and so with body weight, we have to look at, it is definitely multifactorial. While we do have the SNPs that will cause a factor, and they have identified the genes for overweight, we also have to look at environmental conditions as well. So with the adopted individuals and twin studies, they've shown heritability of 75% for obesity. There's definitely an obese gene. But again, lifestyles, including diet and exercise, impact weight. But they're looking at also, are there certain genes that cause a higher or lower metabolism? Are there certain genes that influence hunger more than others?